Everybody's ready to get into biochemistry? All right, so it's amazing how fast the term winds down, isn't it? We have, after today, 4 lectures. Oh, yeah. Forever. Hey, hallelujah, right? Not for me. I do this every year, so it's like, oh yeah. But for you guys, it'll be hopefully the last. Yeah, that's going to say. I'm getting a lot of questions about. Well, we have note cards. This term, I decided not to have note cards. So, no, I don't have note cards. Okay. So yes, we'll have note cards. I will bring note cards next time, and it'll be just like before. You have to get the note card from me, all the blah, blah, blah with that. You have to bring a note card to the exam, all that. Okay? Who said that? That was very effective. I should learn how to do that. That was good. Okay. So today I'm going to finish up saying a few things about translation. And then we're going to move to a topic that is related to both transcription and translation. And it's a broad topic known as gene expression. And so we'll see that transcription and translation both play roles in controlling gene expression. So that's going to be important for us to see. Well, first, last time when I was done, I briefly talked about the fact that termination happens as a result of release factors that come in to the ribosome. These release factors are proteins, and these proteins, there's actually 3 of them. They're called RF1, RF2, RF3. So we have 3 factors relating to initiation, we have 3 factors that relate to elongation, and we have 3 factors that relate to termination of translation. I don't really care that you know the numbers, all right? So there's some that work on UAA, some that work on UAG, and some that work on UGA. The important thing is that the release factors come in when the ribosome is stuck on a stop codon. That stop codon will form no stable base pairs with any of the existing tRNAs. So the release factor has a little section that will form a base pairing like a region with that and cause termination to happen. As I noted last time, the termination happens because of what the release factor brings to the ribosome. It brings a molecule of water, which cleaves the bond between the tRNA and the polypeptide chain. And that polypeptide chain is then released from the ribosome and go out and finish its folding if it's started, or it can go and completely fold if it hasn't already started. So at that point, we're done with translation. There is another ribosome release factor called an RRF. It's actually a little confusing in your book, but there's a separate RRF. And RRF helps to, as I noted very briefly at the end last time, partially disassemble the ribosome. Now remember, all this I'm talking about here is all in prokaryotes. I haven't talked about eukaryotic translation. So we're all talking about prokaryotes. Now you might wonder why partially disassemble the ribosome instead of fully. And the answer relates to the fact that whenever we have operons in E. coli, we have multiple genes. So just because the translation has finished for one gene, it wouldn't make sense to take everything apart if there's another gene right next door that needs to be translated as well. So this is an efficiency mechanism. We don't see operons in eukaryotes, but instead we see something else in eukaryotes that's interesting, and I'll show you that as we get into it. So that's the end of prokaryotic translation and what I want to have to say about that. I want to turn our attention very briefly, and I won't spend much time on it, to eukaryotic translation. So in eukaryotic cells, the process occurs very, very similarly to prokaryotic cells. There are different names, okay? So frequently the initiation factors and the elongation factors will have a little E in front, okay? But you can see this figure doesn't even have them. We're not going to worry about them, okay? The overall process is very similar. We start with the smaller subunit that binds to the messenger RNA, and that makes this initiation complex that ultimately results in the formation of the upside-down snowman. One difference that we see with eukaryotic initiation is the fact that usually the first AUG that's encountered is, in fact, the one That's where translation starts. And as a consequence of this, eukaryotes don't have a Shine-Dalgarno sequence. They don't need something to position, because once the ribosome finds that first AUG, that's generally where translation starts. Okay? Now, as we'll see in a minute, the cap and also the polyA tail play roles in the initiation of translation in eukaryotes. That's also a difference because we don't have those structures in prokaryotic cells. But that's something I'll show you in a minute. Okay. So again, the overall process, basically the same: initiation, okay, elongation, and then finally termination. Now, I mentioned the fact that the polyA tail and the cap at the 5' end both play roles in the initiation of translation. And you can see this sort of set up schematically here for a given eukaryotic messenger RNA. Now, though I don't show you the actual assembly and the, participation of the cap in the initiation, it does play a role in initiation. You can see a eukaryotic initiation factor, that's what that's saying there, 4E, and again, don't worry, we're not going to worry about names here, that is attached to the cap, and it is attracted to a couple of other proteins, EIF4G and this polyA binding protein complex. So what this overall complex does is it favors the formation of a circle. Okay? And you might wonder why it makes a circle like that, and one of the thoughts about why it makes it a circle is that instead of having a long RNA where you have a ribosome that starts over here and it gets all the way over here and then it falls off, if it's in a circle, when the ribosome is done, instead of being way over here, it's not very far away from the place where another round of translation could be initiated. Okay? So the circularization of the messenger RNA probably facilitates additional translation of the ribosome. The ribosome does have to come apart. It doesn't navigate this whole thing without coming apart. So it does need to come apart. But it can find the AUG probably a lot easier if it's finishing close to where the actual start is. Jen?
So, I'm sorry, but is the EIF, the two ones up there, those are the ribosome? Or?
EIF, those are initiation factors. Those are proteins like we saw the IF1, IF2, IF3 in prokaryotes. So in eukaryotes, they usually put a little E in front of it there to indicate it's a eukaryotic initiation factor. And they do the same thing with elongation factors and termination factors. And there are more, I will also say there is a little bit more complexity to eukaryotic translation than what I'm telling you. So there are more proteins that play roles in initiation, like you see these guys here, than we saw in prokaryotic cells. All right? But I don't think for our purposes we need to go into the details of eukaryotic translation at the same depth that we do for prokaryotes. The overall process is the same. Okay, so that's eukaryotic translation. And the next thing I want to say a word about is secretion of proteins. Okay. So far, everything we've talked about, translation occurs in the cytoplasm. You remember in eukaryotic cells, transcription occurs in the nucleus. That messenger RNA has to get processed, and then it gets moved out to the cytoplasm where translation occurs. Well, not all proteins ultimately end up in the cytoplasm. Some proteins get targeted to organelles, some get put in membranes, some actually get kicked out of the cell. So we have to account for all those guys. What happens to all those guys? Well, proteins that don't end up in the cytoplasm start being made on the endoplasmic reticulum. And I'm going to show you how that occurs in a second. But if you look at an electron micrograph, you can see these are actually ribosomes that are on the outer membrane of the endoplasmic, or the membrane of the endoplasmic reticulum. And they form what's called rough ER. They're rough because it looks rough in the image, and the roughness is actually the ribosomes. Those are little ribosomes all over, dotting those little black dots on there. Well, why are they at the endoplasmic reticulum? Well, it turns out that the endoplasmic reticulum, you may recall from last term, is an organelle that is involved in trafficking things in the cell. The The endoplasmic reticulum communicates with Golgi apparatus, for example. It's involved in secretions of things to lysosomes and so forth. And so it's not surprising that the proteins might actually be partly assembled in there. And that's what we're going to see happens. Well, how does the cell know a given protein is supposed to go to the endoplasmic reticulum or to go to the cytoplasm? How does the cell know? Well, it turns out the cell knows by virtue of the fact that proteins that are targeted for the endoplasmic reticulum have what's called a leader sequence near their amino terminus. And that leader sequence generally is fairly hydrophobic. You'll see there's a lot of differences in the actual sequence, but each of these yellow boxes here has a fairly hydrophobic domain following some very basic amino acids. Okay? So that is what is happening. This signal tells the cell this protein is supposed to go to the endoplasmic reticulum. Now, how does the overall process happen? Well, in order for the process to happen, there has to be some proteins and as you saw in the last figure, RNAs that play roles in helping those ribosomes that are translating a protein to make it to the endoplasmic reticulum. Now, I want to emphasize that translation started in the cytoplasm. So we started making a polypeptide chain in the cytoplasm, and that amino terminus came out that something, and we're going to see that something is called this SRP, the signal recognition particle. Something recognizes this and says, oh, look at that basic amino acid followed by that nonpolar stretch. This guy needs to go to the endoplasmic reticulum. So this signal recognition particle binds to it. You'll see it's bound to it here and it's grabbed it. So translation started, and what all this is showing is just a long messenger RNA. Okay. At the very beginning, this signal sequence pops out. SRP recognizes it and says, oh, you're supposed to go down here. So it drags it down to the endoplasmic reticulum. Now, translation is continuing going on while this whole process is occurring. Okay, so there's a receptor on the surface of the endoplasmic reticulum to which the SRP binds. It's not surprisingly called the SRP receptor. And they form an interaction, as you can see here. Notice, here's a G protein again. G proteins play very important roles in translation. These guys are carrying GTP, and GTP is necessary for this process to occur properly. Once everything has gotten docked here, this membrane protein called a translocon opens up. The translocon then accepts the polypeptide chain being made. The polypeptide chain is being made, and we can see it now has elongated from what it was before, and it's starting to appear through the translocon and will ultimately appear down here in the inside, that is the lumen, of the endoplasmic reticulum. Okay? Now, interestingly, adjacent to this translocon is an enzyme called a signal peptidase. Okay? The signal peptidase's role is to cut off that signal sequence. That early sequence that had that nonpolar region with the basic amino acid gets cut off and doesn't make it into the final mature protein. Now the rest of that protein continues being made. Ultimately, the stop codon is encountered, the protein is released, and the ribosome goes along its merry way. Okay, so this is the way then that proteins make it into the endoplasmic reticulum. As you saw last term, a lot of things can happen to proteins in the endoplasmic reticulum. They can get carbohydrate residues put on them, they can be modified in various ways, and then that in turn targets them for specific organelles, the lysosome, export out of the cell, et cetera, et cetera. Okay, I'll stop there and take any questions that you might have. Okay. Moving on. there's blah, blah. You can see more of the movement of various things to other places, as I was talking about before. Okay. Well, the last topic that's relevant for us is actually considering antibiotics. Antibiotics, as you know, are drugs that kill cells. Some are specific for certain types of cells. Other are very broad in their the way they work. Okay, so I'm going to talk about one antibiotic here that is specific for prokaryotic cells, certain bacterial cells, and the other, pyrimycin, is not specific for prokaryotes. In fact, it works on both eukaryotes and prokaryotes. All right. Well, I talk about antibiotics at this point because these antibiotics target translation. Translation is a critical process for a cell. If we inhibit translation, we're going to kill the cell. Just like if we stop transcription, which is what happened with alpha-amanitin, we killed the cell. Similarly here, we're going to kill the cell if we can stop translation. Well, the three examples that I have picked of antibiotics, two of which are man-made, one of which is actually made by a bacterium, target initiation, elongation, and termination. Streptomycin inhibits the formylation of methionine. Remember that formyl group that had to be put onto methionine to get everything, I shouldn't say not the formylation, but the binding of the formyl group on methionine to the ribosome. So if the ribosome can't bind the formyl methionine tRNA, it can't start translation. Because in bacterial cells, that's very specific for bacteria. Eukaryotic cells do not have that formyl group. I didn't mention that. I should reiterate that. Eukaryotes start with methionine. They don't start with a formyl methionine group. That's a big difference between prokaryotes and eukaryotes. Eukaryotes always start with just a plain old generic methionine. Streptomycin, therefore, is specific for bacteria. Okay, so that's a very nice way of targeting. Many antibiotics target specific proteins or specific processes that are unique to bacteria that don't affect eukaryotic cells. And as a consequence, they have very few side effects with that happening. Okay, the next one that I have here is pyrimycin. And pyrimycin inhibits or causes premature termination. And what happens is it looks like an amino acyl tRNA. Pyrimycin fits very nicely inside of the A site of the ribosome. It'll go in there quite readily, but the problem is once it gets in there, it gets linked to the growing polypeptide chain, but it can't be kicked out. And so the ribosome just basically clogs up and stops. Translation is stopped. The ribosome can't get free. You've completely stopped translation in this organism, and you will kill it, whether it's a prokaryotic cell or a eukaryotic cell. This works on both types of cells. And the last one, diphtheria toxin, is important because it blocks the translocation process. And what it does is it actually modifies the large ribosomal RNA. Okay, that's a 28S ribosomal RNA in eukaryotic cells. It covalently modifies it and prevents that modification, prevents the translocation step that happens during elongation from happening. Diphtheria toxin is very nasty. It's a very, very poisonous compound. I was reading up on it just before I came to class. With a toxicity on the order of 0.1 micrograms per kilogram, you're dead. Okay, 0.1 micrograms per kilogram, and you're dead. All right. So very, very nasty compound, and again, it's working on translation. All right. I think that's a good place to stop. Okay. Were there questions on that? I'm sorry, I jumped. That's what it. So it puts an ADP onto ribose. That's right. I'm sorry. Okay. It's time to do some gene regulation. Oh. All right, so gene expression is, and control of gene expression is another type of regulation that we have inside of cells. If you recall last term when I talked about how it was that cells controlled enzymes, I said there were three ways that cells controlled enzymes. All right? One was allosteric control, one was covalent modification, and a third was whether or not a protein was made. Gene expression controls whether a protein is made. It controls how long a protein is around. It controls how long a messenger RNA is around. So all these factors influence the amount of a protein and even if it is present at all. So understanding gene expression is important. Gene expression is controlled not only at the transcriptional level, also at the translational level. It is also controlled by the stability of messenger RNAs and of proteins themselves. So there's four mechanisms I've given you right there that affect gene expression, how much protein is present. Okay? Well, like usual, I'm going to start with prokaryotic systems and say something about those and probably spend the most time on prokaryotic systems because they're the ones that we know the most about. Before I talk about prokaryotes, though, I want to show you this table. This table, I think, is very important for considering the differences, the different needs of cells. All right? On the left column, you see proteins encoded in the pancreas. And by encoded, they mean made, not encoded, but made in the pancreas. And on the right side, we see proteins made in the liver. Pancreas and liver aren't too far apart. However, look at the differences between them. Okay? Pancreas plays a major role in digestion. And not surprisingly, when we look at, and this is the percent of the total protein that's present, 7.6% of the total protein present in pancreas is procarboxypeptidase A1. Well, what's that? Well, that is an enzyme that is breaking down proteins. Important for digestion. In fact, every single enzyme that you see here plays a role in breaking down macromolecules into smaller ones, whether they be proteins, whether they be DNA, RNA, or fats. Okay? On the other hand, if we look at the liver, what do we see the liver has? The liver doesn't have any of these guys over here. Okay? The liver, you may recall, is important in several things. It's important in controlling levels of glucose. It's important in controlling levels of cholesterol. And it's important for packaging up things into LDLs and other complexes so they can go out into the cell and do their thing. In addition, the liver is important for detoxifying things. So the liver's got a whole bunch of functions. It's not surprising we see more even distribution of proteins being made. Okay, here. Looks like it was almost alphabetically made, doesn't it? Coincidentally, but that's just a coincidence, actually. So the needs of these two tissues are very, very different. Not surprisingly, the specific proteins and the amounts of those proteins that they're making are very, very different as well. So we hope to have some understanding then about how it is that cells have this difference and maintain this difference to make proteins as they need them. Okay, well, what I'm going to start talking about is an operon. You probably have heard something about them. I'm moving back to prokaryotes now. An operon, you remember, is a collection of genes under the control of a single promoter. Okay? The lac operon is probably the most studied set of genes in all of biology/biochemistry. The lac operon is a collection of 3 genes that are encoded in the E. coli genome. And the proteins that they code for help E. coli to metabolize the sugar lactose. So that's where it gets its name from, lactose. All right? Remember, lactose, not lactate. Lactose is a disaccharide that contains glucose and galactose. Cells would really like to be able to use that glucose and galactose that's in lactose, but in order to use them, they have to break it down. One of the enzymes, one of the 3 genes that are in that operon, catalyzes the breakdown of galactose from glucose. Okay? Well, E. coli cells don't always have lactose available to them. Okay? Let's say that let's say I'm lactose intolerant, which I'm not, but let's say I were. I would probably be avoiding lactose in my diet as much as I could, and those little E. coli that are in my gut are sitting there going, but please, please, we want the lactose, right? And and I'm saying, I'm sorry, but you're not going to have any, okay? Those guys that are sitting there wanting that lactose, there's no reason for them to make enzymes to break down lactose because the enzymes aren't going to be used. Okay? And in fact, those cells don't make the lactose operon, the lac operon. All right? On the other hand, if I all of a sudden get cured or I take some lactaid so that I'm not going to have lactose intolerance and I go out and decide to have a big ice cream cone, okay? Now lactose is coming through the system. Those E. coli wake up and go, hooray, right? And they want to start making this operon because they want to use that lactose to break things down. So two very different situations, and the cells have to be able to respond to that. So in gene expression, we're looking at the cell's ability to respond to its environment. What are the needs? So two very different needs. One, no lactose. Other, lactose. Very, very simple situation. Okay. There are several proteins that play roles in controlling whether or not transcription of the lactose operon occurs. Okay? One of these proteins is called the lac repressor. Okay? And the lac repressor, as we shall see, binds to a specific sequence near the lac operon. It binds to a control sequence, and we'll see what that is in just a minute. Okay? All right, now getting back. I'm going to skip over this thing. I'll come back and talk about these protein structures in a bit. What I want to talk about is, where's it at? Yes, okay. This is a schematic diagram of the lactose operon. Okay? The three genes that are encoded by the lac operon are called Z, Y, and A. Okay? They have other names, but for our purposes, we'll just call them Z, Y, and A for the moment. Okay? You'll notice that the operon has control sites ahead of it. Okay? And that control sites includes a P, which stands for a promoter. You've already seen promoters before. That's a place where RNA polymerase is going to bind, or at least try to bind. You see another sequence called O, standing for the operator. Okay? The operator is the sequence, as we're going to see, that the lac repressor binds to. Okay? Now, near the operon, notice I said near the operon, and technically this figure is not right because the operon does not include these sequences. Okay, so that really should be right there. Near the operon is another gene called I. It's not a part of the operon. It has its own promoter, and I is the coding sequence for the lac repressor. So the lac repressor is coded for right next to the operon of the lactose operon. Okay? All right. Now, cells make a small amount of lac repressor all the time. Okay? It doesn't take very much. They make a few molecules of lac repressor all the time. And when they do that, the lac repressor is capable of binding to the operator sequence. Okay? Now we call it a repressor for a very good reason. It represses transcription of the lactose operon. It represses transcription of the lactose operon. Therefore, if the lac repressor is sitting here on the O site, the RNA polymerase cannot bind to the promoter, and no transcription will occur. That is, we could think of the repressor as being big and fat. It's covering up part of the promoter. In order for the RNA polymerase to bind to the promoter, it needs to bind to the entire thing. The big fatness of the Lac repressor blocks out the RNA polymerase, and no transcription of these genes can occur. Okay? Everybody with me? Well, cells have to do something besides turn off the Lac repressor. Okay? Because if all they had, or turn off the Lac operon, if all they had was this off system, then there would be no transcription, there would be no making of this. However, I've described two situations to you. One situation where cells have no lactose available to them. That situation I've just described to you. No lactose available to you, Lac repressor binds to the operator. However, when lactose is available, a small amount of lactose makes it into the cell. Okay? A small amount of lactose makes it into the cell. It gets converted to something called allo-lactose, which is related to lactose. And then allo-lactose binds to the Lac repressor. When allo-lactose binds to the Lac repressor cannot bind to the operator. Now, we have a situation, lactose is present, the lactose repressor cannot bind to the operator. Now RNA polymerase has access to the promoter, and RNA polymerase makes the lactose operon, transcribes the lactose operon. Okay, so very simple control system, very, very simple things that it's doing. Okay? Now I'll throw you one more wrinkle. One more wrinkle is the promoter for the lactose operon doesn't look very much like a Tata box. It's not a real good match for the Tata box. What do we know about promoters that don't look like Tata boxes? They don't transcribe very well. They're not very strong promoters, and there's not very much RNA that's made. Well, here's this cell. It's got all this lactose available. This cell would really like to be able to metabolize this lactose, but it's got a promoter that says, make a little bit and we'll be okay. Okay? Well, it turns out that there's another control element. It's not shown on here, but it's actually right up here. Another control element that plays a role. It's called the cap site, CAP. Okay? The cap site is bound by a protein called CAP. CAP has several names. CAP originally stood for cyclic AMP binding protein, but for our purposes, we'll just call it CAP. Okay? CAP, as its original name suggested, binds to cyclic AMP. All right? When CAP binds to cyclic AMP, it binds up here just ahead of the promoter, and it favors the binding of RNA polymerase. Now, we have the thing that the cell needs to turn on transcription at a very high level. Okay? So, CAP acts in a very positive sense. It turns on transcription. Lac repressor acts in a very negative sense. It inhibits transcription. What happens if they both bind? Who wins? The repressor wins. Okay? Because one's binding up here, one's binding up here, and basically the RNA polymerase is going, where the heck am I going to bind? Okay? So when they're both bound, the repressor wins. All right. Now, this operon is, as I said, very, very well understood and has some important implications for people working in biotechnology. And the implications actually arise from this gene called Z. So I want to tell you a little bit about Z. Z has the name beta-galactosidase. B-E-T-A-G-A-L-A-C-T-O-S-I-D-A-S-E. Beta-galactosidase is the enzyme that's coded for by Z. Okay? Beta-galactosidase catalyzes the breaking of lactose into glucose and galactose. Okay? So that's the critical enzyme as far as the cell is concerned for being able to metabolize lactose. These other two genes have other functions I'm not going to go into. If you want to know more, come see me and I'll tell you more about them. But beta-galactosidase is the most important gene for the cell being able to metabolize lactose. Okay? Now, beta-galactosidase, and here's a real good example of this. Beta-galactosidase, here's a cell that has not been exposed to lactose. We look at the level of beta-galactosidase that's made, none being made. We add a little beta-galactosidase, and very rapidly, okay, the cell has a lot of beta-galactosidase there. Okay? So beta-galactosidase turns out to be a very, very useful enzyme, as I'll explain in a second. Before I tell you about that, I need to tell you about one other compound. Okay? And the other compound I need to tell you about is called IPTG. Okay? There's a long name for it. You'll probably call it IPTG like I do. I never can remember the name for it either. Okay? IPTG is a man-made compound. It's a man-made compound. However, as far as the lac repressor is concerned, it looks like allo-lactose. It looks like allo-lactose. Remember that the repressor, when it binds allo-lactose, the repressor can't bind to DNA and transcription can occur. Okay? With this compound, this man-made compound, this is not a cellular compound, with this man-made compound, if I give this to cells, what's going to happen with their production of beta-galactosidase? It's going to start, right? I can artificially induce this. Now, this turns out to be very useful because let's say that I have taken a gene that I'm interested in and I put it in front of the lac control region. If I want the bacterial cell to start making my protein when I tell it to, I put IPTG in there and it starts making my protein in great abundance. Okay? The other advantage of IPTG is the cells don't break it down. They will break down allo-lactose because they use it ultimately. Cells don't break down IPTG. So I give it to cells and it's always going to be in that cell. Meaning that that cell is going to have its lac operon functioning. So as I said, in a biotechnology environment, if I put my coding gene in front of the lac control region and I give that cell a dose of IPTG, they'll start making my protein when I want it to. That's very, very useful. Now, there's another way in which the lac operon is used. I see some frowns. Questions on that? This is the...
I don't know how that works.
I don't know how that works. Okay. All right. All right. Honesty is the best policy, Stephanie. That's good. Okay. I'd much rather know that you guys don't know what something works than to assume that it does. Okay. Let me get back and show you this guy here. Okay. Lac operon, lac repressor binds operator normally when there's no lactose present, right? I've got an artificial inducer of the operon. The artificial inducer is this chemical I've made called IPTG. IPTG looks like allo-lactose to the repressor. The repressor binds to IPTG, and now instead of having transcription turned off, transcription is turned on. Okay? So I have just turned on the lac operon with an artificial chemical. Let's say that I took a bacterial cell and I said, I want to make some human growth hormone. I take and I remove these three genes from this control region. I leave the control region intact, but I remove these three genes and I replace them with the gene that codes for human growth hormone. Now, I've got a human growth hormone gene in a bacterial cell that I give this cell IPTG, and the cell is going to start transcribing the gene for human growth hormone And it's going to start making human growth hormone from it. That's a very common thing that's done in biotechnology. Okay? So I can control when the cell makes a given protein. Yes, sir?
Do you have to add any of the cap protein to make it?
Good question. Do I have to add any of the cap protein to make this thing happen? What I haven't told you is that cap usually is there in abundance, and it usually has enough cyclic AMP that it can bind to the site. So it's usually there, okay? The reason that, so it turns out that the bigger regulator of the operon is actually the repressor. That's why I say if they're both bound, which is what happens usually, no transcription. But when I get rid of the repressor, now I've already got cap there waiting to get transcription started and things take off. Does that make sense? Yes, sir?
What is the purpose of cyclic AMP in bacteria? I mean, we talked about cascades.
Yeah, what's the purpose of cyclic AMP in bacteria? It is not used in a signaling phenomenon like it is in our cells. One of the uses of cyclic AMP is indicating low energy in the cell. Okay. Like AMP is in cells, cyclic AMP can also be in bacterial cells at least, an indicator of low energy. And low energy is going to, again, favor transcription of genes that's going to produce energy. So that's a brief answer to that question. Okay, everybody got that now? Does that make sense? Okay, good. All right. Now, there's a second thing that biochemists have done. I always say biochemists are lazy people that allow them to study gene expression inside of cells. Okay? This involves making another chemical. Okay, if you guys will bear with me on this, we'll sing a song. How's that? Okay. Make another chemical. The other chemical that we make is called XGAL. And XGAL looks to the beta-galactosidase like lactose. The beta-galactosidase recognizes it as lactose. Beta-galactosidase will catalyze breakage of a bond in this molecule, which results in production of a molecule that has a very blue color. Okay? A very blue color. Why is this useful? Well, let's flip the strategy that I just gave you. The strategy I gave you said, let's put a gene in front of this control region and turn on the gene. Let's forget about the control region. Let's take the beta-galactosidase now and put it in front of another promoter of something else. Okay? So I've put the beta-galactosidase in front of another promoter, and I don't know when this promoter is being made. Okay? I don't know when this promoter is being made. And that's not what I want. Oh, I don't see it. Okay, well, let me describe to you. Let's say that I have a promoter that I think is made in the developing embryo in cells that will become fingers. Okay? So I've got this microscope. I'm watching this embryo develop. Okay? And I'm interested in studying the earliest time that this promoter is active because I think it's active in cells that become fingers. All right? So I take this embryo that I've made in the laboratory. Okay, it might be an embryo of a mouse, for example. I guess they have toes. All right? I insert into this thing from square one. I insert this promoter of this gene that I'm interested in, that I think is necessary for making toes, and I link it to beta-galactosidase. Okay? Now, when that promoter is active, beta-galactosidase will be made, and when that promoter is inactive, no beta-galactosidase will be made. Everybody with me? So either I'll have beta-galactosidase or I won't have beta-galactosidase, and I've got a whole mess of cells that I can look at in the microscope. Now I give XGAL to these cells. And I ask the simple question, where is the blue color? Because the blue color will appear wherever beta-galactosidase is being made. And I can say, oh, wow, this is the part that's going to become the foot. I now know that this promoter is active in cells that will become the foot or toes or whatever. You with me? Okay. So the xgal is simply telling me where is beta-galactosidase being made. It also tells me how much. I might see that some cells have a real deep blue color. Other cells have just a little bit. Okay? And when I do this, I can see something about how strong that promoter is in different cell types. So it's a very, very useful tool. So both xgal and IPTG are very, very useful laboratory tools. Okay, questions on that? Yes? Does the xgal kill the embryo so you have to keep doing it over? The answer is, depending on how you set it up, no. You can actually have an organism that's producing that blue color. Yeah. Beta-galactosidase is a pretty innocuous enzyme. Okay, do you guys want to sing a song or do you want to save it for next time? Save it? Okay, we'll save it for next time. Robert, how you doing? Good. Daryl? Hi. How you doing, Jennifer?
I'm sorry.
That's all right. I think I walked into it, so.